Alrighty. Okay, so just to kind of recap here, we've been speaking about the various biomolecules and how they have different structural properties that lend to different functional properties. So far, we introduced carbohydrates. We talked about the monomers, such as monosaccharides, how they can be covalently bound via condensation to produce more complex carbohydrates like disaccharides and polysaccharides. We talked about some examples of those also. And then we got into lipids. Does anyone remember what was one of the properties that we described lipids as being, particularly as compared to carbohydrates? How do we describe lipids? How do we describe lipids in terms of their structural makeup being a little bit different, and that lend to a very important functional property that we see with lipids?
They're hydrophobic.
Excellent, thank you. So they are hydrophobic. In other words, they are non-polar. And we kind of described the chemistry there. We basically said they have less oxygen, and oxygen is really this electron hog. It likes to drag or, hog electrons. And so whenever a molecule has a lot of oxygen in its makeup, we're going to see that uneven sharing of electrons. That's going to give it an uneven or unstable charge. And so that's where we get the polarity that we see with carbohydrates, that we see with water. Lipids are different. They don't have a lot of oxygen, so we don't see this electron hogging or this uneven charge. In fact, lipids have a nice, even, stable charge around the molecule. And so for that reason, they do not play nicely in aqueous environments, and they do not interact nicely with water. And we spoke about some of the benefits of that property when we talk about moving fats throughout our system. We need fats, although they do not interact nicely in the aqueous environment in our physiology, we still need to have fat. And so we can use lipids, particularly phospholipids, to move some of those fats around our cells. We also talked about this idea of the phospholipids as well being an important part of our cell membranes because of their amphipathic nature, their ability to interact nicely with water because of their polar heads. I'll actually jump back here. Because of their polar heads that are facing these aqueous environments, and then there are non-polar hydrophobic tails, which are facing each other. Okay, so we talked about that unique functional property and some of the benefits of that functional property. And let's talk about some of the remaining lipids here. So another class of lipids, a third class of lipids, are called eicosanoids. And basically, we get an eicosanoid by modifying fatty acids. So that essentially, when exposed to certain enzymes, fatty acids can then fold upon themselves. That gives us these more complex ring-like structures that we call eicosanoids. Now, eicosanoids become important with things like cellular communication, cell-to-cell communication. This is how one cell signals another cell to do something. It's also important in things like our blood clotting factors, thromboxane, which we'll talk about a little bit later on, is one of our clotting factors that helps our blood to clot. It is an eicosanoid. And so these are usually derived from really long fatty acids, usually 20 carbons or more, and then they complexly kind of fold themselves to give us these ring structures, okay? Prostaglandins are another or another example rather of eicosanoids, and prostaglandins are going to be important in the inflammatory response and so on, okay? So they have really important functions, but they're simply just fatty acids that have been exposed to certain enzymes that create these a little bit more complex ring-like structures that we see. And then the final category of lipids are steroids. I'm sure we've all heard of steroids. They're a really important they have a really important function in our body. They're all derived from cholesterol. And so basically, these are also ring-like structures, as we can see here, these carbon chain or carbon rings. And these form structures like estrogen, which are important hormones, testosterone, aldosterone, thyroid hormone. Most of our endocrine hormones are actually either cholesterol derived or fatty acid derived, but they're usually steroids in terms of their makeup. So these are also important in terms of reproduction and endocrine function, as you would imagine. And this is our fifth class of lipids, okay? All right. Let's move on now to speak about proteins. So this is our third class of biomolecules, a very, 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 very important class, proteins. Proteins are very abundant throughout the body. Proteins form a whole host of structures. They form other hormones, the ones that are not steroids. They form neurotransmitters, they form receptors, they form a lot of different things throughout our body, signaling molecules, just lots of different functions for proteins and lots of different ways that they can appear in our body. And we'll talk about some of the examples coming up. So they're essentially these polymers or chains of amino acids. So their building blocks are amino acids that are linked one by one covalently. And so this gives us different types of proteins. We've got peptides, which are 2 to 50 amino acids covalently bound to give us that peptide bond. But then we have more complex structures, which are what we actually call proteins. So proteins are really the longer peptides that are more than 50 amino acids long. And essentially, we've got the binding between the carboxyl group of one amino acid. Let's say we have lysine or glycine. We've got a lot of different amino acids. And the amino group of another amino acid. And we can see that exhibited here. So the hydroxyl group, this OH, binds to the amino group of the neighboring amino acid. We release water, so this is also a condensation reaction. And that is going to give us the peptide bond. So it's going to be a more complex chain-like structure. And as you can imagine, we can have many amino acids that really give us the more longer chains, which is what we call proteins. Okay. Now, proteins have different levels of structure. So there are different layers to the structure of a protein. We've got the primary level structure. That's essentially what we just described. The covalent bonds between one amino acid and another amino acid, which are synonymous with these peptide bonds that create this peptide chain or this protein chain. So that's kind of the basic layer or the basic level of the protein structure. The next layer is if we took that peptide chain or that protein chain that only has these covalent bonds that we just described, these peptide bonds. And if we folded this protein into a slightly more complex structure, this is where we get into the secondary structure of a protein. And so basically we see this alpha helix as one type of secondary structure, and then a beta pleated sheet as another type of secondary structure, okay? And again, we're seeing the O on this carboxyl bound to the H on this amino group, and that is how we get these more complex folding of our simple peptide bonds. And then we can get to the tertiary layer of protein structure, or the tertiary level. This is, again, slightly more complex than the primary or even the secondary folding that we just saw. So this is where we now have different secondary structures coming together to give us a more complex tertiary structure. So here we see an alpha helix, which is a secondary structure. Remember, this has that initial peptide chain on it, just folded more complexly into a helical shape. We also see some of the beta pleated sheets that we just described. So different types of secondary structures come together to give us a slightly more complex tertiary level to this protein, or tertiary structure to this protein. And then the most complex level is going to be the quaternary structure of the protein. And so the quaternary structure is where we have, as you would imagine, different tertiary structures coming together to give us an even more complex quaternary structure. And can anyone identify this quaternary example? Can anyone identify what this structure is? Okay, maybe not. So this is actually hemoglobin, and hemoglobin, as we can see, is really 4 complex protein structures that come together to give us one even more complex molecule, which is hemoglobin. And so as we can see, There's a lot of levels or layers to this overall structure. We've got the alpha helical sheets, we've got slightly more complex tertiary structures, and then we've got 4 of those tertiary structures coming together to give us this one hemoglobin molecule, okay? Now I want to point out, let's kind of go back here to the secondary structure. This is where probably the most important layer or level of the protein structure lies. So when we talk about denaturing proteins, remember, if proteins are exposed to excess heat or excess acidity in really acidic environments, they lose some of their structure. And that happens at this level. So denaturing a protein actually disrupts the protein's structure starting from the secondary level. These bonds are broken. And so as we unravel the secondary layer of these proteins, we then, completely discombobulate the entire protein, not only structurally, but functionally as well, okay? All right, I'm gonna pause here and see how we're doing with that so far.
So with primary, secondary, tertiary proteins, they build on each other. Is that how it works?
Yes, exactly.
Okay.
They build on each other. Mm-hmm.
Okay, so a primary is just gonna be like one protein, but then a secondary is gonna be made of 2 primaries?
It's gonna be made of a primary that's folded into a more complex structure.
Oh, okay.
Mm-hmm.
Thank you.
Yep, sure. And then when we get into the tertiary, we can add in different secondary structures, okay? And then as we get into the quaternary, we're seeing 4 tertiary structures here. If we look more closely at each of these tertiary structures, we would see several different secondary structures, which are really just the different folding of that primary structure, okay? All right, so let's kind of keep going here. We're gonna move on to the final class of biomolecules, that's nucleic acids. And these are basically the building blocks of our genetic material, the building blocks of our DNA and RNA, which becomes our proteins. And so we look at the nucleotide structure, there's a phosphate group, which you can see to the left here. There's also a 5-carbon ring centered here, this purple structure, and then that can have a base attached to it. Now, there can be different types of bases that'll give us different types of nucleotides altogether. So if it has a cytosine attached as that base, if it has a thymine attached as that base, or a uracil attached as that base, this is going to be a pyrimidine, right? So a pyrimidine is kind of a subclass of nucleotides that have either cytosine, thymine, or uracil as their functional base attachment. The purines are going to have a double ring structure. So that's going to be the difference in terms of the structure, is that adenine and guanine, which create our purines, are going to have that double ring. And yeah, those are going to be the attachments that give us purines, right? Adenine and guanine. So we can have a lot of different, combinations here that give us different types of nucleotide structures. And then we can have ribose versus deoxyribonucleic acid. That's the difference between DNA and RNA. So deoxyribose just has an additional sugar on there, as we can see here, whereas ribose does not have that additional sugar. Okay, so just a small structural change which gives us a really big functional difference. Let's talk about how nucleic acids form our energy transferring molecules. So there's adenosine, monophosphate, diphosphate, or triphosphate, which gives us AMP, ADP, or ATP. These are the important molecules that help us to transfer energy from one form to the other. We're going to speak about cellular respiration coming up in chapter 3, but essentially ATP is what's made in our body as our currency, if you will. That's our energy expenditure. That's a form of energy expenditure. And it's basically a nucleic acid at the base. It's an adenosine with either 1, 2, or 3 phosphates attached, depending on how much energy is stored in that molecule. If it's got 3 phosphates attached, it's in its highest energy form, which we'll speak a lot about coming up. Okay. We also have nicotinamide adenine dinucleotide, which is NAD, or flavin adenine dinucleotide, which is FAD. These are other energy transfer molecules. We're also going to speak about them as part of cellular respiration. And these can basically shuttle electrons throughout our cells. So NAD and FAD can be reduced. So if you remember, oxidation being loss of electrons, reduction being gain of electrons. So these molecules can be reduced to help carry electrons either into the cell to be a part of cellular respiration, or they can be returned back to pick up more electrons as a part of that electron transport chain. And we'll talk more about that when we get to cellular respiration, but I really just want to drive home that at the core, these molecules are nothing but nucleic acids, okay? We also have cyclical nucleotides. So these are more complex ring-like structures that are formed from nucleic acids. Also important for messengering in our cells, so cell-to-cell communication. Cyclic AMP, we're going to talk about cyclic GMP. These are cell messengers. These can be responsible for being produced inside of a cell, going other places in the cell, and turning on and off other processes inside that cell. So these are really important messenger molecules. And again, they're simply just nucleic acids that are formed into these ring-like structures with phosphates. Okay. Let's practice a few questions, and then we'll move on to speak about enzymes. Okay, so I'm going to launch a poll here. Okay, let's take a look. So which of the following is the most important molecule in the structure of cell membranes? Many persons selected A. Which is correct. So phospholipids is the correct answer. Quite a few other persons selected other options. So carbohydrate, protein, triglyceride. So I really want to stress this. The phospholipids are the most important structure of our cell membranes. If you remember this image from before, we talked about this amphipathic nature of phospholipids, the polar heads facing the water or the aqueous environment, which is either inside the cell or outside the cell, and then the non-polar or hydrophobic tails facing each other, okay? And so by its nature, by its ability to interact with both environments, it's kind of the perfect structure to serve as our cell membranes, to compartmentalize our fluid compartments. Okay, let's look at another question. Final few seconds. Okay. Okay, so a bit of a tie for option A and B. The phospholipid bilayer can easily be permeated, and permeated just means that things can go through by which kind of molecule. And so a lot of persons were between A and B. A is polar and therefore hydrophilic and lipophobic molecules. That's not correct. Let's see what B is. Non-polar and therefore hydrophilic and lipophobic molecules. So neither of those are correct. Let's kind of explain why. So option B is actually contradictory. So if something is non-polar, it's not hydrophilic, it's actually hydrophobic. And if you think about the Latin derivatives of these words, philic meaning a love of or philia of, phobic meaning a fear of or an aversion to. And so a non-polar molecule does not play nicely with water. So it's going to be hydrophobic, afraid of water. Whereas it's going to be lipophilic. It's going to like fats, right? So hydrophobic is synonymous with being lipophilic, okay? Just to explain that, those terms there. So A is not correct, B is not correct because it's contradictory. A is not correct because in order for a substance to go through the cell membrane, it has to be able to exist. Let's actually jump back here. It has to be able to exist in this environment here. Remember, we're talking about a molecule being able to go right through this membrane and make it to the other side. And so if it's a polar molecule, it will not be able to exist in this region here where we have this non-polar hydrophobic fatty acid, phospholipid tails, okay? And so in order for something to go through the lipid bilayer, the phospholipid membrane, it has to be non-polar. In other words, it has to be hydrophobic, meaning that it has a fear of water, it's afraid of water, or lipophilic, meaning that it has an attraction or affiliation to fats, okay? I hope I made that clear. I'm going to pause and see, excuse me, if there are any questions about that. So the correct answer should have been D, okay? To go through that membrane, it has to be non-polar, hydrophobic, or lipophilic. Okay.
So for that question, since the outside is polar, how would a nonpolar molecule go through it?
So it can exist in the cell or outside the cell. So it can exist in the body, but in terms of being able to move through that membrane, it's going to be more favorable if it can interact in this environment. Okay, it's not going to be able to go through, even though it can exist in the aqueous environments. We'll talk about some examples coming up of some of those types of molecules. Okay. Any other questions? Okay. And so just to kind of reiterate that question there, so just to rephrase the question is, if something is nonpolar, how can it be inside the cell where there's all this water or outside the cell where there's water as well? And so the idea is there are things like fats that we talk about that moved around in micelles or other fatty molecules, cholesterols, steroids. They do exist in our environment, but they can go through the cell membrane very easily simply because they can exist between this space, okay? If a molecule cannot exist between the space, it cannot go through that membrane at all, okay? And let's get one more question here, and then we'll keep going. All right, let's take a look. So hydrogen bonding between the amino hydrogen of one amino acid and the carboxyl oxygen of another amino acid in a protein is responsible for which of the following protein structures? Okay, overwhelming response for B, and B is correct, right? That's going to be the secondary structure. So we said, and just to jump back here, kind of point this out while we're looking at this question, we said that when we have the amino group, the hydrogen here, and the carboxyl group, the oxygen here, when we have a binding between these two structures, this is where we get the folding of that initial peptide, okay, or that initial